Hello, and welcome to Cell Truckers. I, the magnificent microscope, shall be your guide today. As you can see in this photograph here, I am a fabulous piece of scientific equipment. Just to tell you a little bit about myself, my specialty is magnification. Or, for those of you sleeping in the back of the room, I make little things appear larger. I am an absolute must when it comes to looking at cells of any form. Did I hear someone say, "What are cells?" Why, they're what you and every other living thing, and things that are now dead, are made out of. Look here, these are cells. As is this. Ooh, and even these. Today, we will be looking at a couple different cells, starting with this animal cell right here. I'll take you step by step through all of the organelles, that is to say, various structures in the cell with specific functions. Let's start with the plasma membrane. This surrounds all cells and regulates what comes in and out. In a plant cell, this would be surrounded by a cell wall. Once inside the plasma membrane, we will be in the chloroplasm, or the area between the nucleus and the plasma membrane. This area is full of cytosol. Cytosol is a fluid substance. We've reached the nucleus. This stores scientific information and controls metabolic activities. On the outside is the double membrane or nuclear envelope. It is full of holes called nuclear pores. In the center is the nucleolus, which makes ribosomal RNA. That mess of DNA around the nucleolus is called chromatin. This coils up to create chromosomes, which are used during cell division. The Golgi apparatus receives vesicles from the smooth ER. It's like a little post office. It packages, processes, and distributes these vesicles within the cell. The Golgi apparatus is also involved in the formation of lysosomes and secretion. This is the endoplasmic reticulum. That's fun to say, you try it. Oh, we'll just call it ER for short. On the right, you have smooth ER, which is the site of phospholipid synthesis, packaging of proteins, and transportation of these proteins to the Golgi apparatus. On your left, we have rough ER. This is the site of protein synthesis and processing. As you can see, it is covered with ribosomes. This funky little guy is a ribosome. They are located wherever protein synthesis occurs. They are made of a large and a small subunit. With the help of messenger RNA and transfer RNA, ribosomes build proteins used throughout the cell. If we travel over here, we'll see a vacuole. Vacuoles are large, but they're much larger in plants than in animals. Oh look, here come some lysosomes to digest the contents of this tasty little morsel. Nom nom nom. Did you say you see a microtubule? You did. This, along with actin filaments, also known as microfilaments, and intermediate filaments, make up the cytoskeleton, which maintain the shape of the cell and causes the cell and its organelles to move. Microtubules also make up the centriole. This is found only in animal cells. Two centrioles, perpendicular to each other, make up a centrosome. This helps to organize spindle fibers. At this time, I will magnify a plant cell. You can see chloroplast to your left. Inside the chloroplast, you can see stroma, a liquid, and grana, stacks of thylakoid discs that absorb solar energy. To your right, you will see the mighty mitochondria. Inside its double membrane is matrix, a fluid, and crista, the folding inner membrane. Chloroplast and mitochondria work in a symbiotic relationship. Together, they make usable energy for the cell. Great teamwork, guys! To wrap up our session, I will enlarge a prokaryote. This is different from the eukaryotes I've been showing you. He uses cilia, the small numerous hairs, and flagella, a large single appendage made of microtubules to move throughout his environment. Eukaryotes also use cilia and flagella. Unlike eukaryotes, though, this little guy lacks a nucleus. Instead, it has a nucleoid. The nucleoid contains a single loop of DNA. Since prokaryotes are only ever single-celled organisms, they do not have cell specialization. Thank you for gazing through my eyepiece into the magnificent world of cells. Bye-bye.